I'd like to welcome you all back. Hopefully you enjoyed the first two speakers, or three speakers actually this morning. I know you're going to enjoy this next one. But before I do the introduction for Mitch, a couple of announcements and commercials. One, right after this session, we will be serving a box lunch, and that's out there. And then across the hall is our table set up, and you can take your lunch and have it in there. When you go out to the lunch, please remember to visit our vendors. They really were a big supporter of this program and of the gathering place. So please make sure you do check out. They've got some great giveaways. And there's a couple good books out there and literature, certainly good for reference that you can have. The commercial I do need to add is that I think most of you are already regulars at Prostate Partners, but if you're not, you'll see in there the literature, the card. This Prostate Partners is the program that is at the gathering place. It is designed for men with prostate cancer and their partners, the caregivers, spouses. We have a list in here. We have programs both east and west, non-COVID, east and west locations that pretty much mirror the same topic. We will vary doctors or that type of thing where that is. So please check that out and watch for that in your email. If you're not getting emails, gathering place, make sure you get that. We also have one that is a program that's prostate cancer. It's an information session that both Bill Imber, where Bill is back there, my partner on the west side, we run that before that program for men that are newly diagnosed as a really very informative session. Everybody in this room has already passed that point, but if you do know somebody, please make sure that they're aware of that. It's my pleasure. I have to tell you that I was diagnosed 7 years ago with my prostate cancer, and since that day I've been dreaming of the day where I can introduce our next speaker. For those of you that know me from our prostate cancer sessions, we always talk about what was your decision factor, how did you make your decision, what to do, who was the advice that you might have gotten. I have to tell you, I've got a great medical team here in Cleveland. Sue's right here, Lee just left. A number of the doctors and Jude is here actually. So a number of my really, really great medical team. But I always said that I had an ace in the hole, I had a secret weapon. And that is my nephew, Mitch Gross, Dr. Mitch Gross, who I will be introducing here. He really is my biggest advisor through all of this and really guided me with a lot of that extra guy that you're always looking for that extra opinion. Well, he was that extra opinion for me. So it was, for 7 years now, I've been waiting for the point where I can bring him to Cleveland and have him speak to this group. So I know you're going to enjoy him. And that right there should probably be enough of a reference that he's my nephew. And I should acknowledge his mother's in the back room too. So his mother came in for this presentation. But now I can give you the official introduction of Dr. Gross. Dr. Gross is an associate professor of clinical medicine at the Keck School of Medicine at USC and the research director of the Lawrence J. Ellison Institute at USC. His academic training includes degrees with the University of California, San Diego, Baylor College of Medicine in Houston, the University of California, Los Angeles, where he got his PhD in molecular biology. His clinical training included residency in internal medicine and a fellowship in hematology and medical oncology at UCLA for the Center for Health Sciences. He is a board-certified medical oncologist with a primary focus on caring for patients with prostate cancer for over 20 years. Dr. Gross, Mitch, is also a physician scientist with broad interest in applying modern techniques related to the study of genes and proteins to the clinical problems faced in treating patients with prostate cancer. His research includes both laboratory and clinical research studies focused on improving care for prostate care patients. And it's great pleasure I introduce my nephew, Dr. Mitch Gross.
Thank you so much. Thank you. Okay. All right, well, thank you. Thank you so much. It's really a great honor to be here in Cleveland and to be able to talk to this group. And the Gathering Place is such a unique center, such a unique program that I hope everyone here in Cleveland really appreciates it, because I know from Los Angeles, I wish we had a program like that there. Okay, so what I thought I'd spend the next 30 minutes or so talking about the genomic revolution of prostate cancer. And you've had a lot of information already today talking about different therapies, lots of acronyms, lots of pictures, and lots of information thrown at you. So I thought we'd take a step back and go back to school here a bit. And let's think about what genomics is. So again, the title is Genomic Revolution. So what do we mean by genomics? So genomics is really the study of all the genetic information in an individual, so all the genes. And remember, the genes are really it's the DNA or the software, the program that tells cells what to do. They could tell the body, tell the cells to grow or to stop growing, to divide, to stop dividing, to move or not to move. So the genes are really the kind of the programming, the software, and they kind of control the functions of life or the functions of cells. So when we talk about the genome, the set of all the genes in a human cell, it's about 20,000 genes. And if you ask, okay, in cancer, how many genes are altered? On average, it's around 4 in prostate cancer. Some cancers more, some less. Prostate cancer, on average, is around 4. So of the 20,000 genes in the human genome, the thing that really starts prostate cancer is maybe mutations in about 4 genes. So let's talk about that a bit more. So in terms of the genomic revolution, it's really a technological revolution. So decades of research starting before and during what's called the Human Genome Project, they basically identified all the genes, the genetic sequence in a normal human person, a human cell, and now through the last decades we've learned all the variations, or we're continuing to learn all the variations. And so really the revolution is widespread availability of this kind of testing that has given us what we call actionable intelligence. So the other key word I want to focus on is actionable genetic testing, testing that we can work with, we can use to determine treatments for cancer. Okay. So the overview of what I hope to talk about again in the next 30 minutes or so is an overview of genetic and genomic alterations that occurs in cancer, especially in prostate cancer. And I really want to focus in on this word, actionable mutations. We'll talk about some examples of genomically informed therapies, but also the limitations and some strategies that researchers like myself and others are developing to try to come out to address this issue of the limitations of this approach. So again, the genomic revolution is really a technological revolution, starting with how much it costs to sequence a gene. So back in the day, 20 years ago, it could cost tens of thousands of dollars to find the sequence of one gene. But over the last 20 years, that price has come down to really almost pennies to sequence a gene. So this is a huge kind of driver for the kind of revolution we're talking about. And what does it really mean in dollars and cents? It could mean that to sequence a gene could be just a couple dollars of chemicals. Now, it costs a lot more to get that result back in a way that a physician can work with and that we can trust. But each test itself, the chemicals, could only cost a couple dollars. And you can even do a whole human genome. You can look at all the genes in a patient or in yourself or a loved one for what's still kind of an approachable level of thousands of dollars. And the typical testing that's done that we'll talk about is in this kind of the retail price might be in the neighborhood of $2,000, but generally again it's covered by insurance. But this is really basically driven this genomic revolution. Okay, so the other topic I want to talk about, again, there's a lot of terms today, is what we call germline somatic mutations. So this is really important as physicians as we approach this issue of genomics, and I want to kind of educate you guys about it as well. So germline means the genes that are passed on. So that's the genes that are in all the normal cells that can be passed on, like from a parent to a child and shared by brothers and sisters in some way. So that's germline mutations. And we have to separate that from what we call somatic mutations. Somatic means the body, so somatic here means the mutations that occur in the cancer itself. So if we have a piece of the cancer, we can look for somatic mutations. Somewhat, we can also look in the blood for this as well. So these are not transmitted. These are the changes the cancer cells basically underwent to change from normal to cancer. So there's somatic and germline. So it was alluded to a little bit, so germline genetic testing in prostate cancer. So this is a figure that represents or shows the kind of mutations if a patient comes in with what we call metastatic prostate cancer. So again, this isn't cancer that's only in the prostate, but this is cancer that has been shown to spread to a lymph node or a bone, metastatic prostate cancer. And we look at the mutations and the specific genes, and the big one there on the right is called BRCA2, breast cancer associated gene 2. There's also BRCA1. It's also called BRCA or BRCA1 and 2. These are the most commonly mutated genes in prostate cancer. About 50% of what we call the germline mutations have that gene. So it's only about 12% of patients have a germline mutation, and about half of those, 6% or so, have BRCA2 alterations. Okay. Now, we'll talk about this more, but these genes that we're taught, that the specific names, and they're all listed there, but almost all of them that we know about are, at least in the germline, are what we call proofreading genes, genes related to keeping the DNA intact. Okay. So now this has now become a routine test, and oncologists and others can order this for patients routinely to see, do you have a genetic risk for prostate cancer? But let's talk about who are the ones that probably really should get this germline testing. These are the inherited genes associated with cancer testing. And we do have these guidelines, they're called National Cancer, the NCCN guidelines. It's sort of like often the payers, because a lot of this also has to do with who pays for it. But so germline testing is really indicated for high-risk patients that have what's called high-risk cancer at the time of diagnosis, or it's metastatic at some point. Those patients, the guidelines now say all of them should have this kind of germline testing. There's positive family history and Ashkenazi Jewish ancestry, also associated with this BRCA1 and 2 mutations we talked about. And when we talk about family history, we mean kind of cancers that occur at an early age. It could be breast or prostate. Early age, generally less than 60, in first-degree relatives, brother, sister, father, mother kind of relatives. But it's not useful in other situations. So one of the things about germline testing is it's generally not helpful in other situations, non-metastatic and non-high-risk prostate cancer. And the important thing about some of this testing is certainly in a patient that doesn't have metastatic cancer, the germline testing is not really actionable. There's no treatment associated with it if we identify a certain mutation in patients that don't have metastatic disease. It has implications maybe for a brother or a father or a son, but for that particular patient, it's not really actionable if it's not metastatic, and that's partly why it's not recommended routinely. Okay. So that gets us to this issue, what we call actionable genetic mutations. So these are the mutations that as medical oncologists, we could really act on. We want to find if they're there because there's certain treatments associated with them. Sometimes we find them by looking at the germline. Again, that's looking at the normal cells. That could be like a cheek swab or a routine blood test. But sometimes we find it by looking at the cancer. So a biopsy or sometimes a blood biopsy, but often just a biopsy of a tumor site. And we send that for this called somatic gene testing or exome testing and other names. But there we're looking also for actionable mutations, changes in the DNA that will have a certain treatment associated with it. Germline testing sometimes can also be done in the blood as well. All right. So what are some specific examples of germline testing? some specific examples of germline testing. So these are some of the companies. There's a lot of commercial companies that do this. Foundation Medicine, Memorial Sloan Kettering Cancer Center, Caris, and Tempus. They all have certain products, commercially available products that can be used.
Okay.
So germline testing, somatic testing. So somatic testing, these are the companies. So, and there's a list of about a dozen companies now that do this routinely, but these are the big ones. So the other point I want to make here is, again, there's 20,000 genes in a human genome. These companies all look at maybe about 500 genes. And these gene testing companies, they're not prostate specific. These are all the genes and all the cancers. They don't develop the test specifically for prostate cancer, so often there are genes there that are not helpful to us as prostate oncologists. Okay. So if we were to kind of look at 100 men with metastatic prostate cancer and try to kind of make sense of their different genetic changes, you might get a graph like this. It's very complicated, but basically these are men going, individual men going as columns, and the individual genes going as rows. So it's not that you can really understand this, but what maybe you could understand is that there's a lot of variation. Many men have different mutations. We talked about fingerprints. This is almost like every man is an individual, every cancer is an individual cancer, and there's certain mutations, changes that are individual to any one cancer. Very few of them are shared across cancers, and the ones that are shared here, for example, would be androgen receptor. That is a very important thing for prostate cancer, but kind of a subject for another talk. The other issue, again, this issue of actionability, only about 20, there's a bunch of these dots down here, but only about 20–25% of them are actionable, that we actually have a drug currently that we can use to treat them. Okay. So I want to really focus on two particular pathways and lesions, genetic lesions that have an action associated with them. And the two in particular are PARP inhibitors associated with DNA repair, what's called BRCA1 and 2. BRCA2 is the most common one. And then immune checkpoint inhibitors, MSI, microsatellite instability, MSI high, and a class of drugs called immune checkpoint. So I thought I'd just want to really fill out for you, I think, this issue. Everyone kind of talks about genetic therapy. Let's talk about what's the sort of science behind it, the mechanism, and kind of what the utility might be. So I kind of alluded to this, that a lot of these genes are DNA repair genes. So there's 3 billion pieces of DNA, 3 billion sequences of DNA in a human cell, 20,000 genes. The biology of human cells have all sorts of mechanisms to protect the integrity of the DNA as it gets passed from one cell to the other, as cells divide. So there's a very elaborate system of protection that's developed. And if something goes wrong in that system, often it can lead to cancer. So what kind of things can go wrong? So how does it work normally? Well, so if you have just something in the environment, radiation or chemotherapy or just bad luck from chemistry, the DNA will have a break. And the proteins that are responsible for fixing that break, there are different proteins in different places. One of them is called PARP, and one of them is called the DNA repair complex. This is effectively what we call BRCA2. BRCA1 is kind of part of this as well. But so they would all come down and find that break and fix it so that the cell can go on living. So this is the normal process that happens. But if you have a cancer cell, what happens is you have a defect in the DNA repair system. So again, this could be BRCA2 could be here, and BRCA1 could be here. It's usually one or the other if it occurs. But so the DNA is replicated, but it does need this other protein called PARP to try to fix that break. Now, if you do what's called a double hit here, or synthetic lethality, if you have a defect in DNA repair and you also have another a drug called a PARP inhibitor that prevents part of that DNA repair system, you have something else occur, which is called catastrophic DNA damage. Then that puts a complete, glitch in the system. Everything shuts down. So if you have a little damage, it's okay. But if you have a lot of damage, the cell has basically self-destruct mechanisms that occur. And it's those self-destruct mechanisms that we take advantage when we treat BRCA mutant patients with PARP inhibitors. Okay. And so this is like a lot of great molecular biology kind of science. What does it really mean to patients? That's, I think, a very important question, especially here. So what I thought I'd do is talk about one of my patients that was treated with one of these PARP inhibitors that we basically presented at a cancer meeting last year, partly because the response was really so amazing. So this is a patient of mine that had a BRCA2 associated prostate cancer that had recurred. So again, this is metastatic prostate cancer that recurred in the prostate area. So we talked about scans earlier. This is called an Oxyman PET-CT. It's one of those special scans that's used in prostate cancer patients. And this big yellow structure here should not be here. This is cancer that has recurred in the pelvis, in the area where the prostate was removed. At that point, again, he had prostate surgery before. His PSA was elevated 1.4. This is what we call recurrent metastatic prostate cancer. And so we did the sequencing testing, somatic testing of multiple biopsies. We actually had to get a total of 3 different tissues. But we were able to find that he had BRCA2 associated prostate cancer, and we put him on a PARP inhibitor called olaparib or Lynparza. And 4 months later, this was what his scan looked like. So you can see a complete resolution of that area there that was in the kind of the left pelvic prostate bed after 4 months of olaparib, and also significant decrease in the PSA as well. So this is kind of the upshot of all that science that's talking about how DNA gets prepared and PARP inhibitors and BRCA1 and 2. It has direct relevance to many patients with metastatic prostate cancer if we can identify the right patients genetically. So actionable genetic test. So the first one we talked about, BRCA2 associated prostate cancer. Second one, I want to just talk about microsatellite instability. Okay. So again, this is another DNA repair pathway. This is a different one, but somewhat similar. What happens in some of the cases where the DNA is damaged, you get like what's called a nick in the double-stranded DNA. It could be, again, radiation, could be sun damage, it could just be bad luck. You get a nick in the DNA. There are these proteins called MSH246, PMS2. They're responsible for fixing this nick because you can't replicate the DNA if it has a kink in it, basically. But if you have mutations, there's certain mutations that basically, gets rid of or avoids that pathway or damages that pathway. What happens is the cells wind up having lots of mutations because now you have basically a broken copy machine. And so the cells will replicate DNA. The DNA will have a lot of mutations. And this is what we call hypermutation phenotype. They'll have lots of new mutations. And that, in a lot of ways, is a very bad thing because then the cancer can learn all sorts of new ways to avoid therapies and to grow and not be controlled. But there is a sort of an upside to this. The upside of this, if you have a lot of mutations, it's easier for the immune system to recognize the cancer cells as being cancer. One of the big problems in prostate cancer is they're not too different from normal cells for the immune system. The immune system kind of sees them like normal cells and then just lets them grow and divide. But if we could teach the immune system to identify cancer cells, prostate cancer cells, that would be a really attractive strategy. And so sometimes, it's only about 3% of prostate cancer, but sometimes they do have that defect where the cells have this mutation phenotype. So this is always kind of complicated to explain, so I kind of reduced it to a little cartoon. So, but what I mean and how we get this treatment is if the good guy is the immune cell, we call it the T immune cell there on the left, and it has a bow and arrow it's trying to attack the cancer cells with. But the bad guy, the cancer cell there, is pink, and it has a bat that's going to basically beat off the policeman, the good immune cell. And so what the hypermutation phenotype, what MSI high or microsatellite instability is it basically teaches the immune cell the wanted poster. It gives us the address to find the cancer cell. So that's a really attractive thing, an important thing for cancer. But so how does this relate to therapy? Well, what we can do is give patients with MSI high prostate cancer a drug that's called a checkpoint inhibitor that basically helps the immune cell in this fight against the cancer cell. And how does that work? Well, so if you imagine the immune cell, the blue cell finds the pink cancer cell. The pink cancer cell has a bat. It has basically a defense mechanism that we call checkpoint that keeps the immune system at bay. And so there's a class of drugs that's called immune therapy or checkpoint inhibitors. There's a number of these, but the one that's used for prostate cancer is called pembrolizumab or Keytruda. So that is the kind of the helmet on the immune cell that protects the immune cell from the defense of the cancer cell and then allows the immune cell to attack. Okay, so this is a big idea, this idea of checkpoint inhibition of immune therapy, or the kind of the negative switch to an immune system that's generally a positive. So this big idea is related to this pembrolizumab, which is approved in 2017. It was approved for any cancer that has this defect. And again, it's only about 3% of prostate cancers, but prostate cancers are one of them. And you don't need to have a certain diagnosis other than this molecular defect. And in fact, this led to the Nobel Prize for Medicine award in 2018 to James Allison and Tasuku Honjo. So this is a big idea. So this whole class of immune therapy has revolutionized many fields of oncology. It's definitely slow to come to prostate cancer, but studies like this and others are kind of helping to increase it. Okay. So really what I spoke about here are the three, what I call the main kind of targeted therapies for prostate cancer. The first one, the top one, is called olaparib or Lynparza. That is a great therapy if you have BRCA1/2 associated prostate cancer. Again, BRCA2 is the most common defect. Rucaparib is another FDA-approved agent for BRCA1/2 altered prostate cancer. So it's the otherwise similar class, PARP inhibitor. And then the very last one, Keytruda or pembrolizumab, that's PD-L1 elevated or MSI high prostate cancer. So we go back to this kind of complicated figure. There's a lot of different kinds of prostate cancer. Every patient's very individual. But the main thing, again, I want to highlight is only about 25% currently have a targetable mutation in metastatic prostate cancer. There are lots of other classes which are up there which we don't have drugs against yet. So this is kind of also very complicated. So now there's databases, even publicly available databases that we can look at to help us link basically a mutation to a treatment. So this is one that's kind of an easy one to use. Basically a company that's out of Memorial Sloan Kettering Cancer Center called OncoKB. So using that kind of database, again, there's about 600 genes that are in the database. They have a certain treatment, about 100 different drugs. Now this is across all cancers, but every day, this kind of database is updated and regularly, and hopefully, people like myself and others, we hope to basically expand the list of drugs and the list of targetable mutations. So what's the kind of the promise of genomically informed therapy is that every cancer has a fingerprint that we can basically use, that practitioners will have, for any mutation we identify, will have a certain treatment, and that these therapies will work very well. They'll have very low side effects, and they'll work for a very long period of time. That's definitely the promise. But kind of what's the reality here? Well, the reality is that, as I mentioned, only about 25% of prostate cancers have a targetable mutation if they're metastatic. So if they're not metastatic, then we really know much less there. But in metastatic disease, only about 25%. Practitioners will have this kind of a long list of treatments, and unfortunately for prostate cancer, that list is only about 3 right now, and that these genomically informed therapies will work maybe forever. And that's not quite true. They work for a very long period of time, but the cancers, even in the genetically defined patients, will develop resistance or could develop resistance. Okay. So how do we get around that? What is medical technology doing to try to get around this issue of the therapy-resistant prostate cancers, or the fact that we don't have a genetic marker for a lot of the treatments we use? So this is kind of one of the latest cutting edge, so I thought I'd just bring it here. So is something called PSMA-targeted immunotherapy. So you might have learned already that PSMA stands for prostate-specific membrane antigen. So this is a protein, basically an address that's expressed on most prostate cancer cells, even if it recurs, even if it's metastatic. And the good thing is it allows the immune system and other systems to target the prostate cancer cells, because most normal tissues don't have that protein, this PSMA. And so it's expressed in almost all prostate cancer cells regardless of the genetics of the cancer cells. And so what we really want to do is use that as an immune target. And this is the strategy, going back to our cartoon here of the good guy and the bad guy. So what's been developed, there's different ways to target PSMA. One way is called bispecific antibody or BiTE therapy technology. So you have basically a chain. On one side of the chain is a PSMA antibody that basically pulls the prostate cancer, the immune cell. To the other side of the chain would be the PSMA. That would be pulls the immune cell and the cancer cells together. So the bispecific antibody is an antibody that has basically 2 sides to it. It's a drug that has 2 sides. One side pulls the immune cell right into the cancer cell, and that's a way to kind of basically target the immune system against prostate cancer. And then this good kind of immune cell there can go on and teach its brothers and sisters, can also educate other immune cells to attack the cancer cells as well. So that's the idea. And so this was data presented last year at a big cancer meeting, ESMO, in Europe, by one of the companies that's making this technology, Amgen, called BiTE antibody. The drug is called AMG 160. So this was kind of presented at the meeting. So it's kind of a complicated slide, but it's that same idea. It's really pulling the cancer cells to and the immune cells together, teaching the immune cells to attack the cancer cells, both directly and then indirectly. This is the data from that meeting. So again, these were metastatic therapy-resistant prostate cancers, regardless of any genetic alterations. In about 30% of patients, there was significant PSA decline. So this was a sign that this is really an encouraging drug. There are many centers in the country that are doing this trial. It's still in phase 1, though. It's still in the early phase, but it's definitely a very interesting technology that hopefully will come to patients, in the next couple of years. So that's really the summary. So I thought I would talk about genetic testing and try to talk about what genomics is and what it's not. We talk about germline testing. That's the testing that gets passed on, genes passed on brothers and sisters and parents and children. Who should get that kind of genetic testing in high-risk metastatic prostate cancer? And then the other thing we talked about was somatic testing. That's testing of the cancer cells itself. And what should we target there? Directly target the tissue. There's about 25% of the time, and currently we have a genetically actionable mutation. Sometimes it's a lot of effort to get to that tissue. It's either a biopsy, sometimes it's blood. The yield on the blood is not as good, but it's much easier to get, so sometimes we do that. And then the specific drugs we talked about are the DNA repair pathway, the PARP inhibitors, rucaparib, olaparib, and then the MSI-high pembrolizumab. And then these other ideas for strategies that wouldn't need a genomically informed therapy, because again, not all patients have that kind of a target currently. That's it. Thank you.
know if we're going to buzz. But
Let's see, maybe I'll stand here.
yeah.
I'll stand. All right. No offense.
It's a COVID.
No offense.
One thought comes to my mind. I remember, I'm old enough to remember Huntington's disease gene and what happened with our insurance providers, you know, kind of blackballing people in a way when that was discovered. But on the other hand, I think about this technology and who wouldn't want to get tested at an earlier time and potentially identify these gene alterations. So what would you say if you had a patient coming to you maybe with localized disease or maybe just metastatic? What would your advice be to when they should think about testing and what tests should they get, germline versus somatic?
Yeah, so overwhelming any patient with metastatic or high-risk prostate cancer in my practice and according to guidelines, I think should have this kind of testing. The most common testing, what we really mean by that is this germline testing. And again, there's a number of companies, Myriad, Ambry, and a dozen of companies that can do that, to look for the germline testing, the BRCA-associated ATM, and some others. The issue can be insurance, but it's part of the NCCN guidelines, and so I don't know what your experience is. Often we can get it funded, or there's like a kind of a clause that the patients only pay a certain amount, and so generally we can get it covered, but it is a challenge. Some insurers, private insurers, are often very, very difficult. But so it would be germline testing. I think the somatic testing really comes in as we want to try to identify therapies, or we really want like a plan B. Overwhelmingly still, BRCA-associated prostate cancer is sensitive to androgen deprivation therapy, which it works so well and for so long it's hard to beat it. But sometimes the cancer can become resistant, and these genetic tests, which one thing we didn't talk about, it takes about 4 weeks often to get back. So if you're really worried about, you want a plan B, it's useful to have that kind of in the record, at least for my practice.
And I would think too for the future, because we're just going to identify, hopefully, more and more drugs that are going to be effective in those certain germlines.
Yeah. So for all those reasons, I think it's good. Now, the germline testing often looks at just about a dozen genes. So, I mean, who knows in the future? But the germline testing is really easy. It's just another blood test. So it's not, except for the expense, it's not really a problem to repeat it.
And I'm sure there's some research, but maybe you could speak on this. For patients that we do see, say, the BRCA2 gene, is there anything coming out that would stop it from mutating so that our current treatments wouldn't work? Are they working on studies in that?
Yeah, so this is obviously a really important area. Is like, if there was something about BRCA2 associated prostate cancer, would there be a different treatment? So in the current situation, the answer is no. We still treat it the same, but unless it becomes metastatic and resistant, then we have different treatments. I don't know. It's like sort of a hard nut to crack. But certainly people, if you look at the breast cancer analogy, people are looking at it after therapy. So patients who have surgery, radiation, other things, maybe BRCA, you would think about giving a PARP inhibitor in certain situations.
Any other questions? I know your stomach is growling, right, for some lunch, so thank you, Dr. Gross. We really appreciate it.
Thank you so much. Thank you.